For the final video in this series, we're going to take a look at what this cell does, this chloroplast does, once it's been able to actually create all that ATP and NADPH using light-dependent reactions. Start by answering these 4 questions on Socrative and then come back. Organisms that produce their own large organic molecules are known as photoautotrophs. Photo because they're using the energy of light, and auto because they're taking the electrons not from other living things or the products from other living things, but from inorganic materials such as water. The special pigments that can donate electrons to a primary electron acceptor are chlorophyll A. And if we had to describe those pigments, I would describe them as strong oxidizing agents because they're capable of forcing the oxidation of water to reduce themselves. And these are part of the reaction center complex because the reaction that they're capable of doing is donating the electrons stolen from water and energized by light to a primary electron acceptor. That then feeds into an electron transport chain used to pump protons, creating a proton motive force that turns the rotor of ATP synthase to make ATP. And at the end, the electrons are donated to that chlorophyll pair in photosystem 1, which are energized by light, donated to a primary electron acceptor, and then given finally to an NADP molecule to produce NADPH. Note that the high concentration of protons is in the thylakoid space. This is the smaller space, and it's easier to create a bigger differential, which results in a greater potential force created by this electrochemical gradient. Now, when donating its activated electron, the chlorophyll in photosystem 2 is a very powerful oxidizing agent. So which of these best demonstrates that ability? Pause the video here for more time. Good. Forcing the oxidation of another molecule is the job of an oxidizing agent. But water does not want to be oxidized, and so it requires a very strong oxidizing agent to be able to force that oxidation. Water is not something that wants to be oxidized, and so this very strong oxidizing agent, this chlorophyll, is actually the strongest oxidizing agent we know. So try these next 3 questions. True or false? Photoautotrophs contain both chloroplasts and mitochondria. Well, chloroplasts are going to be capable of actually generating this glucose, which the cell can use to store as energy. But cells also need to be able to break down that glucose in order to get energy back out again. Photoautotrophs need to have mitochondria in order to be able to do that. And so this is actually true. So even though photoautotrophs need chloroplasts and have chloroplasts to be able to do the job of creating molecules from light and from water, they also need to have mitochondria in order to be able to break them down when they need them for ATP. So true or false, electrons are donated by the pigments of the light harvesting complex, transferring them into the reaction center. This is false. Light harvesting pigments, A, cannot pass electrons, they can only pass energy. And B, the electrons are actually going to be donated from water when it's split by chlorophylls, which are the strong oxidizing agents. And what's the major difference between photosystem 2 and 1? Well, photosystem 2 comes first. Its pair of chlorophyll A's can split water to replenish its electrons, thereby allowing it to participate in linear electron flow, feeding those electrons into an electron transport chain, which then are donated to photosystem 1. Photosystem 1 receives those electrons, those become excited by the light, and then that pair of electrons gets donated away to reduce a molecule of NAD+ to NADH. In cyclical electron flow, there's still no splitting of water. Photosystem 1 is only capable of donating those electrons to another molecule and then receiving electrons from the electron transport chain. In cyclical electron flow, that molecule that receives the electrons is the electron transport chain. So the electrons just cycle back. In linear electron flow, they get donated to an NADP+. But never can photosystem 1 chlorophylls split water. You might have noticed that this process is very similar to the process by which we're actually creating ATP in cellular respiration. The source of electrons is different. The source of energy is different. The actual names of spaces is a little bit different. But ultimately, these two are very similar. There's a chart that's been posted on Web Campus for you to be able to fill out to compare and contrast cellular respiration and photosynthesis. And I would encourage you here to pause and actually try to complete this chart. At the end of the video, I will show you answers. But as you know, the value is in doing it yourself. So pause here and give it a try and check your work when you're done. Now let's talk a little bit more about the Calvin cycle, also known as the light-independent reactions, or dark cycle. These light-independent reactions are going to regenerate starting materials for the same molecules after we're done. It's very much like the citric acid cycle. And in the same way that I would ask you to describe the citric acid cycle by ending where you began, I would ask you to do that with these light-independent reactions. This is an anabolic process. We are going to be building large molecules like sugar from smaller ones. And in order for this to be anabolic, you already know it's endergonic, and we need to provide energy and electrons into the system to be able to make bigger molecules from smaller ones. ATP is hydrolyzed to provide the energy, and NADPH is oxidized to provide the electrons. The fact that we're going to be using so much ATP and so much NADPH to produce even a single molecule of glucose gives you some clue as to how much electrons and how much energy is stored in this molecule and why we can utilize it to get so much energy out when we're done. So the first phase of these dark reactions is known as carbon fixation. It's the actual attachment of carbon molecules to other organic molecules. CO2, a single carbon molecule, is inorganic, but we're going to attach carbon dioxide onto molecules known as ribisco biphosphate or ribulose biphosphate. Or you can certainly call this RuBP for short. RuBP and its attachment to CO2 is catalyzed by the enzyme rubisco. Rubisco, which attaches one carbon of a CO2 onto the 5-carbon RuBP, yields a 6-carbon molecule. We're going to do this 3 times, and the process cannot move forward unless this is done all 3 times. So a single turn of this cycle is going to require the input of 3 molecules of CO2 attached by rubisco to 3 molecules of RuBP. This is carbon fixation. Those 6 carbon molecules are going to be split in half, very similar to the process in glycolysis, where once we had a 6-carbon 2-phosphate molecule, we split it in half to create a 3-carbon 1-phosphate molecule. Here we take 3 of them to create 6 3-carbon 1-phosphate molecule. Now we're going to attach a phosphate onto each end, that empty end without a phosphate, using ATP. So we need 6 ATP at this step to attach 1 phosphate onto each. Now we have 6 3-carbon 2-phosphate molecules. So we're going to use 1 NADPH for each to be able to reduce them. This is why phase 2 is known as reduction. We are adding energy by hydrolyzing ATP, and we're adding electrons by oxidizing NADPH and reducing this molecule. It actually results in 6 molecules of glyceraldehyde 3-phosphate, or G3P. We're going to extract just a single molecule of G3P here. Remember that we put in 3 carbon dioxides, or 3 carbons, So we're going to get 3 carbons out. It takes 2 G3P to become 1 glucose, just like how each glucose yielded 2 G3Ps in glycolysis. So this is only half of what's necessary. The final step is to regenerate RuBP, which is one of our starting molecules. And since we're going to create 3 RuBPs, we actually need to input 3 ATPs, further energy, in order to be able to recreate that 2-phosphate 5-carbon molecule known as RuBP. You need to know RuBP, Rubisco, and G3P. You also need to know that for each cycle, we input 3 carbon dioxides and get out just 1 G3P. And that for each turn of the cycle, we net a negative ATP of 9 and ADPH of 6, and just 1 G3P is yielded. So if you were going to make 1 molecule of glucose, it would take 18 ATP and 12 NADPH. So there's 2 components to photosynthesis, a light reaction and a light independent reaction, sometimes called the Calvin cycle. The light reactions occur in the thylakoids and the thylakoid membranes, where there are photosystems embedded in those membranes. The light harvesting complex is going to take the energy from light, and it's going to use that to be able to excite and energize electrons, which are obtained from splitting water. This is where the release of oxygen comes from. It's a byproduct, a waste product. The plants don't need it. In the stroma of these chloroplasts, the ATP and NADPH, which was made by the light reactions, convert carbon dioxide into G3P, and then this yields a return of ADP and NADP+, which can be utilized by the light reactions, remaking ATP and NADPH from water and light to keep this process going. Ultimately, the energy that's entering chloroplasts as sunlight and from water is what's utilized to make glucose. And sugar, which is made in the chloroplast, supplies chemical energy and carbon skeletons to be able to do other processes. This is catabolism. It's not enough for the plant to make the sugar. It has to be able to utilize it when it's done. And remember that plants can store that excess sugar as starch. At some point in your exam, I'm going to ask you to give me the answer to at least one of these 4 questions. And recall that earlier in this video, I showed you those criteria for how to answer those questions. You should be able to describe why plants give off oxygen gas and why you breathe in oxygen gas, as well as why you give off carbon dioxide and why plants take in carbon dioxide. And it's very important that you explain these in specific detail using your understanding of metabolism. I also want to emphasize that plants do not give off oxygen so that you can breathe, and that you do not give off carbon dioxide so that plants can breathe. It's not all about you. Just like how cellular respiration is something that both you and plants can do, that this process is about doing what's best for the cell. And as one final point, I want to leave you with an understanding that it's important to be thinking about this process in the cellular context as a whole. These gases are being created and absorbed from inside of the cell. They get into the cell, into chloroplasts, into mitochondria by crossing the membranes of those cells. The ATP that's being made is being utilized by the cell to do the kinds of cellular processes that we've been talking about. All of these proteins which are embedded into membranes, those are being synthesized in the ER or by ribosomes that are being found floating in the cytoplasm. The membranes which make up these different components are lipids being synthesized by the smooth ER. The coding for all of these comes from the genomic material in the nucleus, or the genome, the circular genomic material in the chloroplasts and the mitochondria. The fact that in order for us to be able to make these proteins, we have to go through the central dogma from DNA to RNA to protein. which we're going to be coming back to and dealing with a lot in our genetics section. The fact that these products have to actually travel through the endomembrane system, and that's how they're going to get to the places that they need to be, and that all of these components are moving through and across membranes. There are lots of ways in which you can connect this to previous material, and it's important for you to do that. And lastly, I'm going to show you the comparison chart where you can start to compare and contrast cellular respiration and photosynthesis. If you haven't done it, please go do it before you check your answers. But if you have done it, let's see how well you did.